So I was saying that I was going to give you some practical information about the format of this lecture so you can fully participate in it. This talk has an essay format, es un ensayo. The lecture is going to be in English, and you have received by email the full script in Spanish if you need it. We have, as a group, later we are going to hear our names and our backgrounds, but as a group, we have been experimenting for 2 years long on a crossroads between biology and philosophy and aesthetics of the process. Unfortunately, in Latin America, there is an underdeveloped landscape for transdiscipline. This fact imposed on us the necessity to experiment on the margins. Self-motivated, unfunded, we have been recording our discussions. No puedo cambiar la diapositiva con el... Yeah. So we have been recording our discussions to make an archive of our thoughts and our conclusions, and articulating an epistemological framework and various hypotheses that form a certain perspective for biology. Now, this work is both novel and under construction. So there you can contribute to us in the final part with your comments. And we hope this talk brings some new insights to you and broadens your perspective on cellular biology particularly. Transdisciplinary work has the potential to hybridize the codes between different disciplines and thus transform those disciplines, and it has transformed us as researchers. We are based on the work of local and international biologists and philosophers that experience the same aim to assess complexity beyond the regular academic fields or even outside academia with similar challenges to do it. We have reviewed the works of authors such as Karen Barat, Alfred Tauber, Scott Gilbert, Henri Bergson, Gilles Deleuze, and Félix Guattari, Lynn Margulis, Natasha Myers, Carla Hustak, among the most prominent philosophers of the involutionary theoretical approach. I'm going to start with a quote from the founder of the Chilean Society of Cell Biology. And it says, I'm aware that among our colleagues, there are many who view adventures in the history of culture with some skepticism and who do not allow themselves to be diverted from their interest in the hard, so-called hard science, okay? The genesis of the concept of the cell was part of a broader intellectual struggle aimed at displacing German idealism from the study of nature. This makes its history an ideal case for studying the development of ideas in science. I refer to the invention of the cell, because this notion was more a product of imagination than the result of rigorous and critical thinking. The invention of the cell. Wow, a strong statement. Now, critical thinking and imagination are not enemies. So during our work, we have agreed with him in this question, and we propose a perspective that articulates images with critical thinking. To better understand this lecture, my colleague is going to define the two fundamental concepts of our presentation. Daniela.
Hello, everyone. My name is Daniela. The perspective that I'll present to you today is called relational ontoepistemology. For these authors, reality is not constituted by independent entities, an environment where they coexist, like these balls on a pool table. In the manner of the duality of light has wave or particle, object and subject do not preexist their interaction, and the relation cannot be reduced to his part. The nature of the phenomenon changes if we change the apparatus, and the apparatus is the prolongation of a perspective. What exists and how we know are entangled. So I'll define two concepts that they're also approach to the process of the matter: intuition from the philosopher Henri Bergson, and diffraction from the philosopher and physicist Karen Barat. To Bergson, and he said, we refer to intuition here as the sympathy by which one transports oneself inside an object to coincide with what it has that is unique and consequently unexpressible. Analysis, on the contrary, is the operation that reduces the object to elements already known that is common to these objects and to others. To analyze, then, is to express something in terms of what is not. It is important to say to you that intuition is not spontaneity, nor bias or prejudice. It is, in fact, a process against our sensorial and intellectual habits. It is a process of meeting a new perspective that transforms us. And in order to do so, it should transform the terms and dimensions of how we think about it. Knowing the nature of a phenomenon is not about defining its properties, but understanding the array of dimension, how it is presented, always this dimension in motion. Prejudice always involves projecting the self. Bias is a projection of a ready-made point of view. Spontaneity is the projection of a sensitive habit already captured by culture. Intuition is a struggle against our habits. Intuition is not personal. It is not an egoic point of view that I project in the phenomenon. It is a transformative relationship that depersonalizes me because I need to connect myself not with a made object already, but precisely with the coordinates and dimensions where it presents itself that are not my common sense. Knowledge begins as order, not surprise. So intuition is a creative betrayal to ourselves, betrayal to the consensus of representation, and to intellectual and sensitive prejudice. Diffraction, in the other hand, brings us closer to this plane of overlapping. Karen Barat said, diffraction is a methodological approach that seeks to understand how entities and phenomena are not mere reflection of pre-existing conditions, but are entangled emergencies where differences are produced through relations and interactions. It is important to say that for Barat and Niels Bohr, the universe is relational. In other words, bodies are not independent or self-sufficient. They co-constitute interactively. Matter is never settled for Barat. It is materializing in iterative cuts, which he refers to as interactions. For example, unlike particles, which cannot occupy the same space when they meet because they bounce off, the diffraction of waves allows to understand how they can overlap in the same time space. Fixing the cell is precisely the interaction that decides what perspective we are chosen. When we fix the cell, we intervene and make the decision to think the cell has a particle. That is, the cell has a unit. The unit is built by the apparatus, and the apparatus is built by a perspective. Based on this, the following lecture is titled Fixing the Cell, Intuition and Diffraction for a Biology in Motion, and it will be presented by philosopher of process aesthetics, myself, Daniela Céspedes, neurobiologist Oscar Lazo, posthuman philosopher Tomás Flores, and neurophysiologist Nicolás Martinez. Has a choral narration divided on 2 chapters and a short epilogue.
So I would like to start this first chapter, hello everyone, by saying something that may work as a sort of motivation for us to betray the classic concepts. And I hope you will agree with this. Cell biology is in crisis. And many of the key concepts we work with are actively questioned. And we are starting, all of us, I think, to wonder about how they come to be and how these concepts are actually useful. Let me give you a few examples. Let's start by the concept of organelles. Although in the original use of the word organelle, coined by Carl Moebius in 1884, there was no reference to any kind of physical enclosure, we all came to think in organelles as membrane-enclosed objects, and we even study them by using specific properties of the membranes. In the last 10 years, however, we've seen an avalanche of research on membraneless organelles, and I'm sure some of you are into liquid-liquid phase separation, RNA granules, and stuff like that. This idea had always been there, if you think about the nucleolus or the ribosome as organelles, and yet somehow these membraneless organelles were completely out of the narrative. Now, if we focus on the lysosome, as described by Christian de Duve in 1955, as you're seeing in the screen, it is defined by its acidic lumen and also its degradative function. But then very quickly we realized that there were these weird ones that undergo exocytosis. So we started calling them lysosome-related organelles. But then on the screen you see Norma Andrews, who has spent the last 25 years showing us that there's nothing unconventional about secreted lysosomes. That phenomenon happens in every cell they've looked. It is also calcium regulated. It also depends on specific snares. So are lysosomes not secretory devices too? So what are they exactly? And don't get me started on autophagosomes. Those are true multiplicities. What on those organelles would you call functionally homogeneous, or so we can call them collectively autophagosomes? And maybe we can jump into genetics too. Did you know that an impressive 58% of human messenger RNAs have at least one AUG codon upstream to the annotated start of translation? What is what we call a start codon then? And what I want to say is that this is not a collection of misfits. I'm sure it happens in every single research field of ours. I personally work in the biology of neurotrophic factors, and we normally assume that these extracellular molecules bind the cognate surface receptors from where they initiate typical intracellular signaling transduction, like MAP kinases, AKT, that sort of thing. To my surprise, last summer in a conference, I came across the work of Julia Fath, who shows super convincing evidence of BDNF, maybe the most famous neurotrophic factor, containing a cell-penetrating peptide, which allows this classically extracellular signal to seamlessly cross the plasma membrane and directly engage an intracellular signaling pathway. I still, to be honest, I still don't recover from that shock. But the question there is, what is an extracellular signal then? And I think we all process these sort of anomalies as treason. But it's not nature betraying us. You know what they say, traduttore traditore. And that's what we do. We translate our experience of living beings in nature to then formulate theories and laws that will explain this to others. And every now and then, we all mistake our descriptions for the living being. And then sooner or later, I think it's the iteration of something weird happening what makes us realize that we were talking about a concept that we built up at the expense of what we really observed. So when we invented the cell, just to use the same words that Nicolás quoted a few minutes ago, we unified a field. We opened a conversation about cellular life. We started a taxonomy and anatomy of the cell, a physiology of the cell. And in doing so, we necessarily treason our relationship with the one irreducibly complex cell we witness. And singularity is lost in translation. But yeah, at this point, you're probably thinking, well, this singular life, exciting as it may be, it's variable, it's intrinsically irregular, and then it's elusive when it comes to making categorical judgments. So you can fairly ask, and I do myself, how can we build up a valid model to discuss if whatever we study is unstable and variation is the rule? And here is where in his book from 1959, The Logic of Scientific Discovery, Karl Popper makes what looks like a pretty convincing argument about building up knowledge as a consensus. It's a matter of generating falsifiable hypotheses together with a set of shared rules to decide whether our observations support those hypotheses or not. And there you go, universally valid proofs. And a powerful tool of this hegemonic way of thinking is numbers, of course, statistics, which actually came by 100 years prior, when Sir Francis Galton proposed mathematical devices to deal with variation in biology. It was Galton who introduced the median, the standard deviation, the regression line, all those old friends of us. And he actually attempted something that is truly mind-blowing, and I think it reflects very well what we're trying to say here may be a bit problematic. Galton introduced something called the composite portraiture, which is a technique by which he would combine photographs of multiple people to produce an average face. Can you imagine that? So in the 1880s, he actually used this technique to explore the concept of attractiveness, some studies on criminality, ethnicity. I'm sure you find that this sounds just wrong, right? But just like Galton, we normalize the experimental data. We deconvolve the blurry image. We take the background away. In this workshop, you actually get much more sophisticated than that. And when we do so, we build up consensus about functions and mechanisms in the cell, and we localize things to specific compartments. But then always something happens. Sometimes when we have done the experiment many, many times, we start becoming sensitive to individual variation, and subtle anomalies easily stand out and take the stage. So there's a strong paradox here, right? We iterate aiming to normalize, to suppress difference, while as it happens in aesthetics, it's precisely iteration what trains the eye for and makes apparent what is singular in each event.
What perspective can the aesthetic of process give us that the science does not? First of all, memory, assuming that matter has memory of the process, specifically a different emphasis on a scale, time, superposition, and metrics. An aesthetics perspective take us out of the thinking of unity, quantification, and generality. If there is a number at all, it is obscure number. Aesthetics can function transversally, but it will never do so from the discrete number. Therefore, its scale is not quantitative. Its scale is qualitative duration and will seek to increase complexity of the phenomenon. Reality is movement, but this movement is not only exterior, it's also interior. The inner qualitative movement is affection. We understand affection has a trace of memory, the echo of a relation. Every cell affects and is affected. As we are not able to separate the subject from the object in an independent way, we suffer from the object, and this object transforms us when we want to apprehend it. We suffer from the cell, and when we fix it, we produce an affect and create it in that perspective. Thus, the cell is both objective reality and the relation with the apparatus of production that affect it. Cell is not just an abstraction. Let me give you an example from geometry. The circle does not exist without being drawn. Even if this drawing was made virtually by our minds, the circle is always a roundel movement, but the circle is the abstract fiction of an unaffected roundel. A circle has no memory. The roundel does. Representations require emptying the phenomenon of affects. They take from it the affective memory. Affections are the echoes, as I say, of a relation. From this perspective, when we encounter matter, we always find it towards its materialization, already affected by relations and creating new affection. So matter is both creator and agent. If we know a singular relation, we know it by its affection. In contrast, science functions propositionally. It must designate a state of affairs, create representational functions, so that even if its focus is in tendency of the phenomena, it will be from statistics. Because it has to distinguish a states of affair, creates points of view, assigns a metric of those points of view, and aspects that are not groupable under that point of view are seen as a negativity, as an anomaly, or a noise. Science in that way is always incomplete because it multiplies points of view to infinity. So there can be as many cells as their points of view. But aesthetics is what is qualitatively in between these points of view. In relation, this is what we are going to call interstices. They are in between. They form a movement perspective. Because for aesthetics, the anomaly is not an anomaly of reality, it's an anomaly constructed by the point of view itself. Qualitative continuity between the points of view is what we are going to call intuition. Usually, science uses intuition in its hedonic experiments, which can only remain mental. Aesthetics resist representation, homogenization, and above all, and this is important, transforming layers of time into an instant, because since the instant does not speak to us of that accumulative quality of contrast, it does not speak to us of the memory of the process of matter. Interstices are singular relation of expression, a cumulative time of memory that expresses itself and adds a nuance difference to reality, which did not exist before. Obviously, a scientist can be an aesthete. They are not mutually exclusive. Etienne Sorieux, who is one of the pioneers of process aesthetics, presents a beautiful hypothesis. He says, things exist, but they lack of reality. What can an existence lack to become more real? To increase in strength, in extension, in consistency, are these vital singular elements that provide inspiration, that inspire our interest. Perhaps it probably makes sense to you that it is singular that moves the intelligence. Now, Darwin was a scientist and also an aesthete. According to the researchers Carla Hustak and Natasha Myers, Darwin engaged in a sensory relationship with his experimental subjects. In the absence of a sucking insect, Darwin introduced himself into the pollination scene to demonstrate how orchids actively modify their anatomy in response to insect activity. Darwin assumed the roles of both pollinator and pollinated. Experimenting with the space of more modes, he used his finger to mimic the movements of an insect. His experiments led him to fully engage on a sort of a role-playing in which he used his body and his weight of instruments to mimic the movement of insects with a pen, with a brush, or even the tip of a pencil. In short, the aesthetics links metrics and codes without homogenizing them. It feels a phenomenon with quality and memory. It feels with time and history. It assumes that nature is creative. Nature creates its own non-human codes. It does not dichotomously separate nature from culture. Aesthetics requires intuition, since for every phenomenon, the relationship has to modify our terms in the encounter and violate, as I said, our sensitive habits. For each aesthetic intuition, we have to betray our prejudice.
And I would like to jump in just to emphasize that I don't think we're saying anything you haven't already experienced yourselves, which is this tension between two different modalities of betrayal. In order to communicate, we treason the uniqueness of the relationship with the individual organism, especially when it doesn't fit the model, just because we need to put it in terms that we can discuss with the community. But also, we treason the consensus as we let the experience of iterative observation transform us back and create something new, something rich, something where we don't exactly know how to communicate.
Well, this treason that Oscar was talking about, or I may say confusion, could be particularly diaphanous if we look at the history of how optics have influenced our construction of ideas about the cells. The first optical observations humans did of what we today call cells were through chromatic lens in the 16th century. Those lenses do not focus all the light wavelengths on the same planes, so you will see the same cell multiplicated like in several focal planes. So it's interesting that despite people have never seen cells before, they thought that those were not the right conditions to study cells, okay? So let's stop here to think about that. It's an important point where we can comprehend epistemological bias of representation expressed as an optical criterion. What's behind this idea that those were not the right conditions to study cells, that we are in front of an improper representation of reality? What's behind this attitude that biology cannot be studied as a diffracted image? So Alfred Tauber say, theory and technology act reciprocally. We build those technologies that we believe are important to examine a particular perspective of nature, okay? Later on, during the 1700s, movement patterns on fresh microscopic samples ultimately lead to the consensus that what was being observed under the microscope were like animals of smaller size, okay? And this is also interesting because the analogy of cells as small animals shows that researchers already thought that there was a microscopic environment that can be studied just as we experience the macroscopic world. That is not obvious. It's an assumption that we can study microscopic world in the same rules of representation that we study the macroscopic world, okay? So in the macroscopic world, we have the possibility of a complex immersive experience. We feel, we think, etc. In other words, a situated perspective. On the other hand, in the microscopic world, to have this immersive situated perspective only through optics, through the microscope, is not obvious. So researchers inadvertently adopted microscopy as a method to study cells that intrinsically diminishes the intuitive approach by leaving out aspects of the sensorial experience. Later on, in 1826, apochromatic microscopes were invented, and they solved this technical problem, okay? Those improved lenses show single cells as single defined objects. This technical modification allowed researchers to observe cells as they thought it was proper. Many prejudices, and as we conceive them today. Consequently, cellular limits, membranes, gained a lot of attention. And here we are now, until now, in the era of membranes, okay? Based on membranes, generalizations about cells started to being assumed, up to the point where cells emerge as the gold standard of the living. We have the theory of the cells with its postulates to define the living. based on what has membranes or not, more or less. In this context, neurons were the last step for generalization about cells due to the technical difficulties to find their limits, okay? You know, to distinguish neuronal physical limits due to the arborization and Baroque morphology. Santiago Ramón y Cajal explicitly say in his book, Neuronismo or Reticularismo, Neuronism versus Reticularism, that he and his contemporary research groups discarded hundreds of samples that suggested continuity between neurons because they were already looking for the limits, okay? We can make a third stop here and analyze this again. Inadvertently, the notions about biology of cells based on optics were already operating to reinforce an epistemological and technical pathway that fixes, delimitates, and constructs partial models. In brief, this early history of cells already had the elements of their contemporary representational rationalization. Everything that is hardly delimitable is considered optical noise, and thus epistemological noise. This jumped the thresholds that we put on our microscopy at experimental designs to conclusions about the cells in general is dangerous. But if we redefine the limits between the thresholds and the noise, between the valuable information and mistakes, another cellularity could emerge. We affirm that this hegemonic perspective gravitates towards representational epistemology at the expense of intuitive and diffractive epistemology. This perspective, that is adequate to physically and theoretically delimitate cells from one another, and just then constructs models of group of cells, tissues, animals, also supports the still ongoing race to improve techniques to study cells as the main way to comprehend physiology and of health and disease. So there is a technical race to improve techniques that is based on this bias about cells also.
This vision centered on the individual supposes a progressivism because it thinks that advances in discoveries are equal to a more specialized knowledge, that is, more focalized, more directed to units. In that sense, our current vision of the cell is a reflection of the liberal vision of the human individual. An alternative way of thinking could signal to us that with every step further in the way of the individualization of realities, something hides itself, like when we are in a swimming pool trying to reach a ball and the ball escapes further away because of the waste we ourselves produce. But if nature hides itself, is not by a limitation of our senses, it's because it's not made to be sensible exclusively for us. It's because it creates a diversity of points of view more than be given to points of view already created. Nature follows the principle hide to create. Important part of what Bergson says in the first chapter of his book Matter and Memory is that we tend to perceive things in a specialized way, meaning in a division in fixed parts, limited by a relatively rigid border because it serves to our practical interests. We literally perceive the world according to what we can physically grasp or virtually manipulate with our hands to carry out an action, and we leave outside that which doesn't interest us for that action. That's why Bergson says that perception, contrary to what had been thought in the philosophical tradition up to that point, doesn't illuminate things, but rather darkens and obscures them. In other words, in a universe that is already light, those things that are not of interest to our actions become dimmed. Thus, perception is construction. It's an invention which is based in the creativity intrinsic to nature. The same goes when we approach cells as a production of nature. They are in itself multiplicities, not units. There is not a cell in nature, there is a cell just for the human eye, because of the opacity we introduce even with the microscope. That's why, in effect, we see a cell, and that's what needs further explanation. How is this configuration of limits created? How come we see a figure, we see a ground? One answer is that there is a process of differentiation that is intrinsic to nature in a way that figure, although it distinguishes itself from the ground, carries the ground with it. Philosopher Gilles Deleuze shows this using the image of the lightning. The lightning, says Deleuze, distinguishes itself from the black sky, but must also trail it behind. And he adds, it is as if the ground rose to the surface without ceasing to be ground. That means, as we have already said, that relation is first, and not the units, even when one of the units is thought as ground. Then, what is a human abstraction is the independence of the figure from the ground, not its differentiation from it. The point of start is the object of what Daniela said. We identify something that doesn't fit with our current views and schemes of thinking, and we call it an anomaly. That's the diffractive experience with what was first seen in the microscope. From there on, we engage in the almost moral practice of tagging that anomaly as something negative that needs to be corrected. Because we confuse reality with what we are used to see. We confuse reality with what's intelligible, legible for us. And that's just a part of existence produced by our immersion in the worlds to which we choose to pay attention. The diffractive experience with the microscope was, and still is, the first sensible modality of a cellular world becoming important to us, a cellular world beginning to matter.
And I think at this point, it may be useful to make a connection with the phase contrast microscope, something I'm sure we're all familiar with. Phase contrast allows us to see precisely not the specimen itself, but the relationships between the specimen and the light it scatters. So we see constructive interference, which we emphasize even more by optically dimming down the background. So it would be fair to say that it is the specimen making itself visible to the microscope optics, right? And your active role, my active role, is setting the instrument to let it emerge as a legible entity, but the visibility is provided by its own interaction with light. During the last years, I've been blown away by holotomography, as a similar interferometric device that uses laser to bring the same concept to high-content 3D live cell imaging. So there you go. Those are diffractive devices among us.
For Bergson, to intuit the singularity of a melody from its abstraction is almost impossible. It is like expecting an alien to guess our way of speaking from written language. However, it is not definitive, since every representation has a vestige of a rhythmic intuition. For example, Stephen Hawking manages to embody and understand the logic of a black hole without having had any direct perception of the phenomenon. Even without knowing it perceptually, he can intuit it through mathematical abstractions and representations. Intuition is at the level of immediate data, but mediation can also lead us to a rhythmic ecology. Returning to the question of this chapter, the button is the organized representation of an abstract plane of an immanent melody. But in each representation, there is an echo of this immanent melody, an echo that is not an object, but of affective relations. Affect is what allows us to distinguish how that which exists becomes important, acquires value and strength within the phenomenon. But we know affectively. We know by giving value in one thing and not to another. In this exercise, we make agential cuts and we suggest a tendency of the phenomenon. In aesthetics, we treat this tendency in a qualitative way for a living body, and this is what the father of ethology, von Uexküll, will define. It is always an ecology of signs, ways of attracting and repelling, as Darwin did through the iteration with orchids. We'll understand rhythmic has a logic of coordination between heterogeneous space-time. Intuitive work make us sensitive to this logic of transduction and generative transformation between dimensions. Another example, Isaac Newton became sensitive to gravity from the perpendicular effects and affects that this force has on the body of the apple. How it is that Newton came to formulate the law of gravitation inspired by such a mundane event as the fall of an apple? In William Stagley's memoirs, the author mentions that Newton tells him, why does the apple descend perpendicularly towards the ground? There must be a power of attraction at the center of the earth. There is a force that extends through the universe. Deleuze and Guattari created in 1980 the concept of multiplicity in 1,000 plateaus, which will replace the concept of unity. If representation homogenizes a phenomenon to make it common from social terms, intuition makes it common from the singular. In other words, it can only become common to us if it transforms us. To define multiplicities, multiplicities are not defined by properties. We know a multiplicity by knowing its n dimensions. These n dimensions can be qualitative or quantitative, or both combined. We already know that intuition allows us to think in both metrics, even if they are contradictory. What defines the dimensions is always the research question, which makes a cut and exclusion in the subject matter. For each question, a different multiplicity appears. In 1,000 plateaus, Deleuze and Guattari give the example of coins to understand that two questions construct different phenomena. I'm going to explain it with the same x, but different multiplicities, that is, with different n dimensions from the two different questions. If I want to know how many coins I have in my hand, I only need one metric. This set of coins in my pockets forms a numerable, discrete, and finite multiplicity, 5 or 4 or 12. But if I change the question, for example, and I want to know how many places and with what variations of temperature these coins have been in a certain period of time, I have a continuous and non-numerable multiplicity, which Riemann, the mathematician, would call coordinate points and not elements. So Riemann says that this continuous multiplicity appear in the colors, for example, in the chromatic spaces. That is, we cannot directly compare the parts, and we require the construction of a criterion of translation. This in aesthetic, it's called a meta-metric of what Bergson would approach with intuition.
To go forward, giving you another example, I'm sure you are familiar with the work of Lynn Margulis, the one who proposed what we now know as the endosymbiotic origin of eukaryotic cells. That 1967 work of hers is super interesting to look at because she understands, for the first time ever, evolution, not as the process of how units are transformed, but as the becoming of multiplicities. And she identifies the encounter between cooperative organisms, bacteria in this case, as the driving force of evolutionary change, rather than transformation, individual transformation. The story is later picked up and brought further by Gilbert, Sapp, and Taube in an ambitious work called A Symbiotic View of Life. We have never been individuals. And what they do is, starting from the idea of the holobiont as the assembly between the host and its microbiome, they challenge every aspect of animals' individuality, genetic individuality, because genes can jump across species, immune individuality, because immune systems develop in cooperation with the resident microbiota, and also physiological individuality, because microbes are essential for some of our physiological functions. So crucially there, the self doesn't appear by building a wall that delimitates it, but in the contextual dynamics of living as a multiplicity. Now, I obviously have my own criticisms about whether or not these ideas fly well as biological theories, but what is absolutely crucial from this case is how these notions challenge our research practices. I don't care about the conclusions these researchers arrived at. I mean, but the question is super interesting. What is the cell you're studying? How it becomes that? And most interestingly, how you manage to study it in a way that makes it appear, differentiate from the background, and show not its properties, but its dimensions. Tomás, are you there?
Until this point, we haven't answered how it is that, as we said before, we effectively perceive something differentiating itself from the ground. Now we can say that what differentiates itself is not a unit, but a multiplicity. And how can we experience multiplicity? With intuition. Intuition, as Deleuze defines it, reading person, is the sensible apprehension of difference as multiple processes of becoming. And the relevance of this for science is not only epistemological, but also ethical. Or at least that's what Karen Barat says is involved in the ethics of the scientists. That is, being responsible for what is left out in the ground, in a way in which we could say the ground rises to the surface without ceasing to be perceived as ground. Contrary to when the noise is left behind in microscopy because it was not considered relevant as information about the cell, or the effective statistical infinite probabilities that are left behind when the isolated result is communicated in a paper. We have to be responsible for the fiction we call knowledge, and that's the role and the importance of Bersonian intuition for the sciences. In which ways we come to perceive and to feel difference, and what are the epistemological, ethical, and even ontological implications of this open multiplicity of sensation. We also said that there is not a cell in nature. Now we can say that there is, but only in the relationship between a cellular multiplicity and the human who observes or isolates one part of that multiplicity. That relationship is also a product of nature. Even when there are microscopes involved, the microscope prolongs a natural body and transports the visual system of that body to the microscopic world of the cell. The microscope is a way in which the body can go beyond itself, or what philosopher Gilbert Simondon calls transduction, that is, a connection between heterogeneous sensible worlds. That's what was involved in what Daniela said when she spoke about a coordination of dimensions that precisely are heterogeneous and incommensurable between themselves. But still, they can produce a relationship in which something becomes sensible as a nuance, or as Bergson will put it, as a mixture. That's to embody a black hole or to capture the gravitational force by the sensible encounter with the apple. Now, to complete this picture, we need time. There is an example chosen by Bergson of a lump of sugar, which, if it is grasped solely in terms of its spatial organization, we do not grasp its singularity as a phenomenon. But if we approach it in the process of dissolving itself, its way of being time is revealed. In that scenario, human action, for example, the spoon with which we can dissolve the sugar, is just one more component in what Bergson calls blocks of space-time, that is, moving blocks in which the tool is not a mediator, but it enters as one more sensible element in that world, what we have been calling affect, a world that creates itself at the same time that is constantly differing from itself.
So the hypothesis that this intuition that Bergson has defined can become a method to understand nature has been already proposed. Okay, in our group we have a diverse perspective regarding this. Let's start by saying that when our assays give us results that are hard to frame on our working models, kind of two options emerge. Either discard them as unuseful mistakes or noises, or intuition, okay? The reaction that we have to deal with those mistakes or the noises in the microscope, and also the scarce sharing we do about our intuitions conditions our understanding of nature. If a theorist can give us an homogenized perspective on a problem, on the other hand, its testimony can tell us about its emotional atmosphere and its embodiment. I'm pretty sure every one of you has notion of intuitions in the context of your research. We, as a group, think that those notions should be shared, should be taught to students, should be studied, and could prolong up to a certain methodology for cell biology. They are valuable. The testimony is also epistemology. From our analysis, certain critical tensions arise. Maybe you are thinking about them too. How would instruments, microscope, change if we thought about them more intuitively? What could hypotheses become in this way of understanding research? How should the education of a researcher that incorporates intuition be? How will we integrate the already known about cells with the unavoidable perspective of intuition that it's on between us, like a ghost haunting us? Which scientific research community could arise, and how should we communicate science? We invite you to the answers. But I'm going to mention a couple of highlights we have found. We are approaching the end of the presentation. But first, as Daniela said, intuition is not about having a spontaneous personal feeling or a spontaneous perspective for each one. That will make a methodology impossible. Intuition is an active effort that recreates itself in front of every research topic. The material expression of this will be to slow the research process down and generate particular languages and methods to each phenomenon. Second, since intuition is synesthesia, it will tilt the communication of science toward the communication of images more than concepts, synesthetic images. And third, transdisciplinary work, okay? That is to say, experimental designs and instruments that allow us to oscillate between representation and intuition. This may not be entirely assessing what Bergson meant for methodology of intuition, but it could serve us as a tool to start softening our representational thinking, incorporate intuition to biological science, and also find it when it's already operating. Now, if you tell me that this proposal are impractical, for example, to slow down the process of research, tell me why. Why this is impractical. So a final question can be, what it is structural in our environment that constantly inhibit us of thinking biology in a different way?
As we have already seen, multiplicities are defined by the dimensions involved in the mode of presentation of the phenomenon. And these dimensions will be defined in virtue of the question we ask in that moment. We have seen that one way of asking this question is the one that creates a membrane as a way of fixing the cell. But another way of asking the question does not allow us to think of a membrane, or at least not as a rigid border. A question generates an agential cut in matter and leads us to specific dimensions of it. The key distinction to challenge a mode of thinking centered in units is to say that it is not enough to multiply points of view already given, but that it is necessary to situate ourselves between the singular differences that create points of view and traverse their movement. That is what we will call perspectivism, and we will end this talk characterizing this mode of thinking to then talk about ghosts in biology, and finally, why it is important to let ourselves be affected by the singular culture of cells.
Boccioni, the artist of futurism, criticized Picasso by comparing him to scientists, saying that he was an analyzer of fixity, that he copies the object in its formal and fixed complexity, not in the form of movement, making it incapable of experience itself in its action. Boccioni said, Hasbergson, by stopping the life of the objects, it analyzes it and kills it. The above is precisely a vision of Cubism as a relativism. Relativism could be, in the model of unities, a generality that affirms the supposed difference between particular points of view already individuated. On the other hand, perspectivism is something else. And actually, I explained it with Picasso himself. When Picasso was painting nudes and drawing women lying down, this is a painting from 1941, Picasso starts with the classic scheme, left arm bent, then he changes perspective and the image of the left breast and the right leg seen from the side, crosses with the back through the sheet. So far, Boccioni is right, we have a simple relativism of units, but what Picasso does in 1941 is to incorporate the movement of his own body on a painting. Then we no longer have isolated points of view, but we have what we call this here, a perspectivism, the intrinsic and qualitative difference between the points of view in movement and becoming, which come together in a different way of seeing the unity, which is a process of contrast and memory.
So maybe a question is, how can we move around cells and incorporate this movement? And just as Tomás was suggesting, I keep thinking about permeable BDNF and how it shows the emergence of cells in diffraction. If I look at receptors at the cell surface, permeable BDNF is unimportant for the question. Membrane trafficking keeps being absolutely crucial to localize signal at specific membranes. But if I turn and look at BDNF, it is the membrane what becomes irrelevant. And now it's all about how BDNF distributes so it reaches different targets in time and space. In this cell, as a multiplicity, both things happen. Both perspectives coexist prior to any attempt to study them. And they create different topologies of the same cell, different perspectives and different ecologies. And that's super interesting. And then when we think about the devices we can use to explore and create this sort of cell culture in broad sense. I remember this other ghost. A few years back, I met Tama, an expert in phantom limbs, and she has shown that phantom limb pain is an expression of how the brain creates a continuous experience of our body, even after part of it has been removed. So in the absence of the arm itself, for example, the peripheral activity of the remaining nerve endings shape an updated map in the brain. And by this diffraction, we project a very vivid perspective of an arm, even with no arm whatsoever. And there's something quite powerful here, I think, which is that we actively create ourselves a body using whatever we have in hand. So if borders and limits of our body are not strictly spatial, we may be able to expand ourselves into it and use this sort of body flexibility, natural body flexibility, as a device for scientific endeavor. microscopes, nanomachines, optical tweezers. Could they be a way into cell culture? Our way to meet cells halfway through?
The diffraction and the epistemology of intuition open up new questions, assuming that we are a part of the nature we seek to understand. And that is, understanding is not external to the phenomenon, but interactive. Our knowledge practices are generative. They create cuts and perspective. If we include an articulative and qualitative mode, allowing ourselves to be affected by the culture of the cells, I mean with this, to be affected by the unique ecology we establish with each one of the cells, it could inspire us to construct new criteria. This could allow us to creatively reconfigure how we think about life and the cells. As Alice Fulton says in her poem, because truths we don't suspect have a hard time making themselves felt, as when 13 species of whiptail lizards composed entirely of females stay undiscovered due to bias. Against such things existing, we have to meet the universe halfway. Nothing will unfold for us unless we move toward what looks to us like nothing. Faith is a cascade. Thank you.